Morning. Today's topic is going to be transcription and translation. It is Sunday morning. The Bears are playing the Saints at 3:30 today. Go Bears. I am not hopeful, but you never know. That's why they play the game. So hopefully they can pull out a miracle here. But I'm not getting my hopes up. So today's topic is transcription and translation. That is protein synthesis, right? So we're going to be taking information that's stored in DNA and using that information to guide the production of proteins inside of the cell. So we're going to go through, there's two stages to that. The first stage is transcription. The second stage is translation. And so we're going to go through how transcription works and then how translation works in this video here today. So let's go ahead and take a look first at RNA, which is the type of genetic material that has a big role in protein synthesis. It did not have a role in DNA replication, right? DNA replication occurs before a cell is ready to divide. It's just taking DNA and making a copy of it. So RNA is not involved there. However, it is an important nucleic acid in protein synthesis, right? And RNA stands for ribonucleic acid. There are a few differences between RNA and DNA. One of those differences, RNA is single-stranded, not double-stranded, so it doesn't have that double helix structure. It is single-stranded. It contains the nitrogenous base uracil instead of the base thymine, which you can see over here. It says uracil U in RNA, thymine T in DNA. It contains the sugar ribose instead of deoxyribose. So you can see they are both pentoses here. They're 5-carbon sugars. The big difference is on the number 2 carbon in deoxyribose, there is just a single hydrogen atom, whereas on the number 2 carbon in ribose, there is a hydroxyl group. So ribose has that oxygen. Deoxyribose does not have that oxygen. Okay. And RNA is found both in the nucleus in eukaryotic cells and in the cytoplasm in eukaryotic cells. Of course, prokaryotic cells don't have a nucleus, so we don't say that about prokaryotic cells, but DNA, on the other hand, is only found in the nucleus. And another thing about RNA that I don't have listed here, but RNA is shorter than DNA because RNA is simply used to make a copy of a short segment of the DNA molecule that just has the instructions for building one protein. And that short segment is called a gene, rather than copying the entire DNA molecule. So RNA is much, much shorter than DNA. Okay, so let's take a quick look here at the flow of information by comparing a prokaryotic cell to a eukaryotic cell here. In prokaryotic cells that don't have a nucleus, all of protein synthesis takes place in the cytoplasm. The DNA is floating around in the cytoplasm, technically in an area called the nucleoid region. And that DNA is then transcribed or copied into a form of RNA called messenger RNA that contains the instructions for building a protein. It's the copy of a short segment of that entire DNA molecule with the instructions for building one single protein. And that messenger RNA, or mRNA, is then translated into a polypeptide by a ribosome, right? Because we know that ribosomes make proteins. They make proteins using the instructions in messenger RNA. And this all takes place in the cytoplasm. In fact, the ribosome may attach to the messenger RNA and start translating it before the entire messenger RNA molecule is done being transcribed. So it's kind of hanging off of the DNA molecule while the end of the mRNA is being transcribed, and the ribosome actually attaches to the free end and starts translating it before the messenger RNA is even completed being built from transcription. In a eukaryotic cell, however, transcription, the production of the messenger RNA, takes place inside of the cytoplasm. And before that messenger RNA leaves the cytoplasm, it gets slightly modified in a process called RNA processing, which we will take a look at in more detail later on in this lecture. And then once that processing is done, of the pre-mRNA molecule that is made, or the primary transcript is what this is sometimes called also, that final completed messenger RNA will then exit the nucleus through nuclear pores, and translation occurs in the cytoplasm. Okay, so there is a little bit of a difference in protein synthesis between prokaryotes and eukaryotes. We're going to mostly focus on what takes place in eukaryotes today. So Francis Crick of Watson and Crick coined the term the central dogma of molecular biology, because that's really what we're studying right here and now is molecular biology, right? The study of DNA and RNA and protein synthesis, that is molecular biology. And what he said was that DNA can be used to make a copy of itself, and then also transcribed into RNA, which is then translated into protein, right? And so that is the central dogma or belief system of molecular biology about the way that things work. Now, we do know there are other types of RNA besides messenger RNA, right? So this isn't the complete story. Messenger RNA is one of the types of RNA that is made using DNA as a template. And that messenger RNA is then translated at the ribosome into protein. But we also know that there's ribosomal RNA or rRNA. We know that there is transfer RNA, which is tRNA. There's microRNA. There is small nuclear RNA. There's all types of RNAs. And every different type of RNA that's found in the cell is all produced through translation. Just not all of it is that, or through, I'm sorry, all of the RNA that is produced in the cell is produced through transcription, but not all of it is then translated into protein. Ribosomal RNA is used to build ribosomes. Transfer RNA, tRNA, is used to bring amino acids to the ribosome to put them together to make a protein, right? So there's different types of RNAs, and those different types of RNAs have different functions in the cell, and all RNA is made through transcription. Messenger RNA is the type of RNA that then gets translated into a protein. So the genetic code, the instructions that are coded within the DNA for building proteins is encoded as a triplet code. So every 3 bases is the instructions for another amino acid to be brought to the ribosome to be added to the protein. Okay, so the question is, why does this genetic code have to be a triplet code? Why can't it be a single base code or a double base code, for example? All right, so I want you to think, how many different amino acids are there in the human body? There's 20. Okay, so there's 20 amino acids. So if we just had a single base code, that there are 4 different bases, right? In DNA, there's A, G, C, and T. So there's 4 different bases. And if we just had a single base code, that means that one code would just be A, which could only code for 1 amino acid. Another code would just be G, and another one would just be C, and a 4th code would just be T. So you'd only have 4 different possible codes. Well, that's not enough, because you need 20 codes since there are 20 amino acids. Why not a double code? Same concept, same logic here. If you've got 4 bases arranged into groups of 2, how many different ways can you arrange those 4 bases in groups of 2? Well, that's just a math problem, right? 4 bases arranged in groups of 2, how many different combinations can you have? 4 to the second, right? Which is 16. Still, that's not enough. Different combinations for the 20 amino acids that exist in our bodies, right? So the triplet code, if you think about it, 4 to the 3rd power, if you have 4 bases arranged in groups of 3, how many different ways can you arrange those 4 bases into groups of 3? 4 to the 3rd power, which is 64, which is enough that you would be able to code for all 20 amino acids. And you're going to have extra codes, so you're going to have duplicate codes for the same amino acid, right? So if you look at this code here, and this is the genetic code in RNA, right? So this chart here is a codon chart. So it tells you if the codon were uracil, 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 that would be the instructions for phenylalanine to be the amino acid put in place. at that point in the protein. But UUC, uracil cytosine, UUC is also a code for phenylalanine. Okay. And you can see ACU, ACC, ACA, ACG, all four of these are all codes for the same amino acid, which is threonine, right? So these are the three base codes, and this is the abbreviation then for an amino acid in this codon chart. So then the question is, okay, but why not quadruple or quintuple base codes? That would also give you enough, right? If you had 4 to the 4th or 4 to the 5th different combinations, that would also meet the requirement of having enough different combinations for 20 amino acids. All right, but there's several reasons why. If you had quadruple or quintuple base codes, the DNA molecule containing the instructions would have to be much, much larger. So your cells would have to be bigger to store them. And we know because of the surface area to volume ratio, concerned cells have to be small. Both replication as well as transcription and translation require energy. And the bigger the molecule that you're building, the more energy the cell would have to expend to build those different molecules. You're also going to, if you have to put more bases in place, there's more potential places for mistakes to be made. So your mutation, the number of mutations that you're going to get is also going to be larger, right? The amount of time it's going to take to build new proteins and to replicate DNA would also be much longer, right? So there are several reasons that would work against selecting for nature having selected for quadruple or quintuple base codes instead of just a triplet code. So a triplet in messenger RNA, a 3-base sequence with the instructions for 1 amino acid in messenger RNA is called a codon. Okay. One really cool thing is about the genetic code is that it is universal. So it is the same from one organism to the next with very few exceptions, which means that you can take the gene for one protein in one organism, put it into another, and that organism with the new gene will be able to express it, which means it will be able to transcribe it and translate it. Okay, so we could take the gene that leads to the production of the enzyme that allows fireflies to produce light, put it into a tobacco plant, and now the tobacco plant can produce light. We can take the gene out of a jellyfish that allows it to bioluminesce, put it into a pig embryo, and that means that all the cells that descend from the ones that picked up that gene in the pig embryo will express that gene. And so now the pig can bioluminesce. Now, we can't just stick it into an adult pig and have the entire pig start to bioluminesce, because only the cells that you injected it into would have that particular gene. But if you put it into a cell in the embryo, then all of that cell's descendants will have that same gene and will be able to bioluminesce. Okay, so why is the genetic code being universal important? Well, that is more evidence for a common ancestor for all life on Earth, because all life uses the same genetic code. So they probably had a common ancestor that used that code, and it was passed down through all the billions of years of evolution on the planet. Okay. All right, so how does transcription occur? Okay, transcription is a 3-step process. There's going to be an initiation, elongation, and termination, right? So now we're talking about how is mRNA made. Okay, transcription begins at a DNA sequence where the enzyme that carries out transcription actually binds, okay? And the sequence of DNA that the enzyme that carries out transcription, called RNA polymerase, where it binds, is called the promoter region, okay? And promoter regions can be recognized by a specific structure called the Tata box, okay? No giggling, okay? The Tata box is a sequence of thymines and adenines at the beginning of a promoter region, okay? And so The first thing that's going to happen to initiate transcription of a particular gene is that special proteins called transcription factors will bind to the promoter region. Okay. Once that happens, the specific enzyme that we're looking at here, called RNA polymerase II, will bind to the promoter, and that forms what's called the transcription initiation complex. Okay. Now, the second stage of transcription is the elongation of the messenger RNA. So once the RNA polymerase is firmly attached to the promoter, the DNA starts to unwind. Okay, so this doesn't require helicase or anything else here. The RNA polymerase attaches, the DNA unwinds, and RNA polymerase II will start then attaching complementary bases to the template strand of the DNA. Okay, so we have a template strand that is being used in transcription. The other strand that's not being used is the non-template strand. RNA polymerase II will work at a rate of about 40 bases paired up per second. And what's interesting is multiple RNA polymerases can bind to the same gene simultaneously. So one attaches, starts transcribing, another one attaches behind it, starts transcribing. Because when a cell needs, say, a particular enzyme produced, it doesn't need just one copy of that enzyme made, right? It's going to need thousands of copies of that enzyme made. So how is the efficiency of transcription increased? Well, you don't have one RNA polymerase attach, transcribe the gene, then go back to the beginning and go again, and then go back to the beginning and go again. You have one attach, start, and another one attaches and starts, and another one attaches and starts. So you can make transcribe multiple copies of that same RNA molecule very, very quickly. Okay, now, what signals the end of transcription? How is it stopped, right? Because we don't want to transcribe the entire DNA molecule. We just want to transcribe this one particular gene. So in prokaryotes, there is a transcription termination signal at the end of the gene that is reached, and the RNA polymerase just falls off the DNA. Okay. In eukaryotes, there is what's called a polyadenylation signal sequence that is transcribed. Okay, so in the DNA, that's going to be TTA. It gets transcribed into AAA. When that sequence is transcribed, the RNA polymerase will continue going for about 10 to 35 more bases downstream from that polyadenylation sequence, and then proteins will cut the transcript free from the RNA polymerase. Okay. Now, like I said, in prokaryotes, the RNA polymerase falls off. That messenger RNA is actually oftentimes start to be translated into protein before the RNA polymerase is even done making the messenger RNA molecule. But in eukaryotes, remember I mentioned that the messenger RNA that is made is called a primary transcript, and that actually has to be processed. It's not ready to be translated yet. It has to be processed before translation can begin. So what happens with the primary transcript after it's made? The first thing that happens is what's called a 5' cap gets added. And that actually happens before transcription is done. It happens after about 20 to 40 nucleotides have been transcribed. A modified GTP molecule is attached to the 5' end of the primary transcript. After the transcript is cut free from the RNA polymerase, enzymes add what's called a poly A tail to the 3' end of the primary transcript. Okay, and so what that means is that enzymes add anywhere from 50 to 250-ish adenine nucleotides to the 3' end of our primary transcript. Okay. Now, the question is, why are these things done? One, it helps to get these out of the nucleus. And two, there's a lot of enzymes inside of our cells that degrade RNA very, very quickly. So when they recognize the RNA molecule, they degrade it, which means they break it down. And the 5' cap and poly A tail help to prevent that from happening. Okay. This also helps the GTP at the beginning, helps our ribosomes to attach to the 5' end of the messenger RNA molecule. Now, you might notice we see here it says 5' UTR and 3' UTR. UTR stands for untranslated region. Okay, so where the RNA polymerase bound to the promoter, remember where that ta-ta box was located, there's going to be a sequence of DNA nucleotides that are transcribed that are not actually part of the instructions for the protein. They're not part of the protein code found within that DNA that gets transcribed. However, there will be one particular codon that is a start codon, that is the beginning point of where translation will occur at the ribosome. And then there's going to be another stop codon where translation will end at the ribosome. However, there are bases transcribed after that stop codon. And so we have this 3' UTR as well. All of these bases that were transcribed after the stop codon will not be translated either. Okay. The final piece of RNA processing that occurs in eukaryotes only, this does not happen in prokaryotes, is what's called RNA splicing. Okay, so your average eukaryotic gene is about 27,000 base pairs, okay, which will then also be the length of the average primary transcript. However, the average protein is about 400 amino acids long. So for 400 amino acids, how many base pairs are needed? Well, each amino acid, the instructions for one amino acid requires three base pairs, right? So if we have 400 amino acids times 3 base pairs per amino acid, we would need 1,200 base pairs for the instructions for your average protein. So if the average gene is 27,000 bases long, but the instructions for your average protein just need to be 1,200 bases long, that there's a whole lot of extra information in there. Okay, so there are actually long non-coding regions within each gene, found within each gene, that are called introns, which stand for, that comes from the fact that they are intervening sequences. And then there are coding regions that are called exons. And you can think of exons, you can help because they exit the nucleus. They are actually what remains after this RNA splicing, because those the exons are what the parts of the gene that contain the instructions for our protein. Okay, so RNA splicing is done with RNA splicing. Introns are removed, and then the exons are joined together. Okay, there is a short nucleotide sequence at each end of each intron that is a kind of a signal for splicing, which is carried out by small nuclear ribonuclear proteins called SNRPs. And I always think of them as like these little blue molecules that are moving around, because it reminds me of the word Smurf. They're not actually blue, but just kind of, that's the first image I get in my head. And these SNRPs, these small nuclear ribonucleoproteins, bind to these sites within at the end of each intron, right? So small nuclear ribonucleoprotein, right? They're small. They're found in the nucleus. They contain RNA and protein combined together. That's why they're called small nuclear ribonucleoproteins. And these are what bind to those sites at the end of each intron. Okay, so here is your SNRP, right? With the small nuclear RNA and protein, okay, combined together. So this is one SNRP. And many of these will join together, okay, with other proteins in the nucleus and form a structure called a spliceosome. And the spliceosome will then cut out the introns and join the exons together. Okay, and so then what we get, the cutout intron is released. Enzymes will degrade these small segments of RNA here, so that the RNA nucleotides can be recycled and reused in the nucleus. The exons are joined together, And what exits the nucleus is our finished transcript that has only exons spliced together with a 5' cap and a poly A tail. So I have a couple links here that I will share in Schoology so that you guys can see that RNA splicing in action. Okay. Now, interestingly, the RNA is the catalytic portion of a SNRP. Okay, it's not the protein, right? When we think of anything that catalyzes reactions inside of a cell, we usually think protein, right? Because enzymes are proteins and they're catalysts. But with these SNRPs, the RNA portion of the molecule is actually the catalytic portion. So we would call these ribozymes. Anytime RNA acts as a catalyst, we call that RNA molecule a ribozyme. How can RNA act as a catalyst? Well, RNA bases can pair with other bases on another region of the molecule, so they have a specific three-dimensional shape. Some of our RNA bases contain functional groups that can participate in catalysis, just like the functional groups of amino acids in the active site form weak, hydrogen bonds and hydrophobic interactions and ionic bonds with their substrates. We can have the same thing here. We have functional groups on these RNA bases that can form these weak interactions with their substrate in order to catalyze a reaction. For example, in the adenine molecule here, we see the amino group right here, right? We've got hydroxyl groups down here on the sugar. So we've got all these functional groups that are capable of forming these weak interactions like hydrogen bonds with their substrate. Okay, so RNA can hydrogen bond with other nucleic acids, and so there is some specificity there. Okay, so why do we have introns, right? Like, why would we have these intervening sequences? All right, there's a couple of reasons. One, a single gene can be spliced differently, which produces different proteins then. That's called alternative RNA splicing, right? So so here's our primary transcript, sometimes called the pre-mRNA, right? That has been produced through transcription, and it's got these different sections of introns and exons. And we can take different segments of that primary transcript and cut them out and join the remaining segments together in different ways, which leads to the production of different proteins. And those different proteins then will carry out different functions in the cell. When the Human Genome Project was done, it was known that humans had about 100,000 different proteins in our bodies. And it was believed that we were going to find that we had 100,000 genes also, that we should find that there are 100,000 genes. Well, it turns out that the actual number of genes in our bodies is somewhere around 20,000 different genes. So those different 20,000 genes have to be able to be, their instructions have to be able to be put together in different ways to build different proteins then. Okay. In addition, we can have different exons that code different structural and functional regions or domains of a particular protein. Okay, so here we have a gene that has 3 different exons, and maybe one of those regions, say this is a membrane protein, right? That's going to have a receptor region that is outside of the cell. It's going to have a region that anchors it in the membrane, and then it's going to have a catalytic portion on the inside of the membrane, right? And so we could have one exon that codes for the catalytic portion of, or enzymatic portion of the membrane protein that would be on the inside of the cell. Another exon that codes for the region that is embedded within the membrane and anchors that protein in the membrane. And then you could have a third exon that encodes the portion of the protein that is the receptor part that's outside of the membrane. Okay, and then finally, when meiosis occurs, which is the type of cell division that leads to the production of gametes, we can have the shuffling of exons when crossing over occurs. So 2 chromosomes can exchange pieces, so they can then exchange exons with one another. So we get the mixing and matching of exons of different genes during crossing over, which could lead to the production of new proteins, which those new proteins could be beneficial. They could have no impact on the cell, or they could be harmful. We don't know, but it's a way to generate more variety in the offspring. Okay, and so that is where I'm going to stop this recording. This is the transcription recording. I'm going to do translation next.